new session, and I would like to invite you to the new disease, which we are closing the discussion about SMA, and now we are going to Duchenne muscular dystrophy, which is also has a lot of challenges and new coming treatments, and also new coming I think barriers, and not only access for the treatment but also generally in the system, which is I think very similar in SMA in some points. I would like to welcome Professor Francesco Muntoni, Pediatric Neurology from the United Kingdom, who is also from Paediatric Neurology and Development Neurosciences Department, Institute of Child Health in United Kingdom. Welcome, and I would like to also say that the professor will present of us genetic therapies of Duchenne muscular dystrophy. Welcome, the floor is yours.
And I hope you can hear me well.
Yeah, we hear you very well. Welcome.
Okay, I believe I need to put the slides forward, and that you will be putting the slide at your end, and I will be, that's correct, and I will be making them advance. Well, thank you. Firstly, I was listening with interest to the discussion regarding SMA and newborn, and I hope and I wish we will be in the Duchenne field in the same position as Duchenne, as Panamuscular atrophy is in a few years to come. So we'll talk about the Duchenne muscular dystrophy genetic therapies. So the click works, that's fantastic. And these are my disclosure. I'm involved in a number of clinical trials that I will be discussing. And I also involved in some of the companies' advisory board, as you probably expect for people who are involved in many of these clinical trials. And I declare this conflict of interest now. And I will be talking about the impact that novel therapies have for Duchenne, but I will be focusing on the genetic therapies. I will not talk about the pharmacological therapies. And I will be largely talking about the exon skipping for out-of-frame deletions, because this is the field that is evolving more rapidly. I will not talk about nonsense mutations. This is there are also therapies there. Ataluren is a drug that has now been around for quite a while. So in a way, I take that as an established fact as opposed to novel therapies. And I will also take you through what's happening regarding the gene therapy for Duchenne. I think the first point to discuss is the, in people with Duchenne muscular dystrophy, the majority of the mutations are out-of-frame deletions. 65% of people with Duchenne have this type of mutation, and another 10% approximately have duplication. I mentioned that there is one particular therapy for nonsense mutation, that is Ataluren, that is available for approximately 10% of the patient. This is a drug available in Europe. And then there are a number of other smaller mutations that are at the moment a little more tedious to detect, but I think also the diagnostic actually there has moved quite a lot. So it's possible now to find these other small mutations much better than just a few years ago. And regarding the Duchenne muscular dystrophy, I indicated that the majority are out-of-frame deletions. What that means is that the reading frame of the gene is not maintained. And you need to think about the reading frame as this slide where there are the numbers from 1 to 79 that are the different exons of the gene for Duchenne muscular dystrophy. And some of these blocks, as you see, are light blue, and they are rectangle. And if you take away one of those, for example, 35, you take away 35, you can see the 34, 36 match well. So that would be an equivalent of a deletion that does not affect the reading frame. But if you remove the number 6, for example, you can see that number 7, number 5 don't match well. So the frustration is that in the majority of people with Duchenne, just one of a few of the exons are missing. The majority are there, but because you've been removing an exon that has that affects the reading frame, you end up with no protein. And the protein in this patient is, let me just go back one. Oh, it's okay. Yeah. So the protein in muscular dystrophy is the dystrophin protein is this red rim that you see around every single muscle fibers, and on the bottom you see a black box, and that black box is what will happen if you take a biopsy of an individual with Duchenne where there is no dystrophin. I think the nature has already given us some information of what could be done to try to exploit the fact that the majority of the exons in people with Duchenne are still there. And this experiment in nature is Becker muscular dystrophy, that is a much milder form, is still a muscular dystrophy, so there is progression, but compared to Duchenne, it's much milder. And in that condition, also there are deletions, but as you can see in that particular example, exon 45 and 46 is missing, and 44 and 47 match well together. So you end up being able to produce some protein, as the bottom panel shows, with some light red staining that is much better than nothing. And therefore, a number of companies have been identifying strategies to try to change the way the mutation is handled by the body. Not changing the gene, this is an important point, but changing the way the gene is processed to make the messenger RNA and eventually the protein. And in the example you have here, this will be a patient with a deletion of exon 49 and 50. As you can see, 51 and 48 don't match. However, if you remove with antisense oligonucleotide exon 51 and you induce a splicing of 48 to 52, you end up with a Becker-like molecule. And I think this is what has been done with antisense oligonucleotides. So to create a Becker-like molecule, I think that the important aspect of this is that this is not permanent. These are antisense oligonucleotides that need to be given regularly, every week, by intravenous infusion. And my group has been involved in the development of the antisense to skip exon 51 at Pilsen. And more recently, I've been involved in developing another antisense for exon 53 called Golodirsen. However, there are other efforts as well. There is a Japanese and American effort to do similar antisense for exon 53 called Vilto-Larsen. And a company called Sarepta is developing an antisense to skip exon 45 called Casimersen. I think if you like, there have been a number of the one that I just mentioned are the efforts that I indicated on the slide in the bottom in red as morpholino antisense oligonucleotide, because you can have different chemistries to synthesize to make this antisense oligonucleotide. Many of you are aware that other antisense have been developed in the past. And I think it's important, this is an important lesson for all of us, that the therapy development is a complicated business, and as much as we all like successes, there can be failure as well. And in the past, a company called Prosensa that partnered with GSK and Biomarin developed an antisense to target exon 51 and also 45 and 53 and 44. But eventually those efforts did not proceed, while because of not enough efficacy and some toxicity as well. And more recently, although I'm not going to talk about it, another company called Wave also developed an antisense to skip exon 51, but the program was terminated last year because of lack of efficacy. I think the antisense in the bottom, the morpholino, have been better tolerated. And there have been evidence of efficacy, initially from some of our trials here in the UK, but then mostly by trial in the States by the group of Jerry Mendel. And I think these, if you like, this led the Morpholino to go forward, and more recently, the addition of a Japanese company that is also developing the Morpholino. And I will briefly give you some high-level sense of what can be achieved, and it's important to be realistic. These are not a cure for muscular dystrophy, okay? But I think you induce the expression of low level of dystrophin protein. You have on this slide on the left-hand side, this particular patient, 01015, at baseline with no protein recognizable, and after treatment, there is recognizable protein. Many fibers have dystrophin, many fibers don't have dystrophin, and the levels are certainly not normal. And despite, if you like, the fact that you do not end up with a normal level of dystrophin, there is effect. This effect takes time to be developed, and I think this slide gives you a sense of the number of children in the blue who lost the ability to walk over a period of 4 years in a particular clinical trial. And you can see that already in year 1, 17% of those children stopped the ability to walk, but they remain constant in terms of number, while the number of children in the who did not receive treatment, who were matched for inclusion-exclusion criteria, over a period of 4 years, a little by little increase that percentage of number of children who stop walking up to 83%. So this is some indirect sign of efficacy that is also been, if you like, strengthened by some more recent data to indicate that the respiratory function at the later stage, because many of the people in the upper panel at some point will have stopped walking anyway, but if you continue to follow these people up, you realize that the respiratory function decline has been nearly half compared to what would be expected to occur year on year. So it's not a cure, but is a way to slow down disease progression. And to some extent, we're finding something very similar for the antisense oligonucleotide to skip exon 53. This is we published recently the data related to the expression of the protein in this patient with a biopsy at 48 weeks with unequivocal expression of dystrophin you could visualize on Western blot, on immunocytochemistry, and on molecular biological techniques. The level are relatively low, but despite this, and these are a patient, I hope you can see it on the left-hand side before treatment, on the right-hand side after treatment, with a number of fibers that have clear expression of dystrophin. But I think the most important thing that everybody really wants to see is not low level of dystrophin, but what is this dystrophin doing to patient? And this is yearly decline in 6-minute walk test over a period of 3 years in a group of patients who match for the inclusion-exclusion criteria of children in our trial. And this, as you can see in this group of children after 3 years, the loss of distance in a 6-minute walk test is of 181 meters. While in the patient who received the treatment, there is also decline, but this decline is of 99 meters with a difference of 82 meters between the treated and children who did not receive the treatment. And along similar lines to what you saw with the eteplirsen, there appears to be a relative slowdown of the condition in terms of, for example, the number of people who stop the ability to walk. And you can have here in gray the people after 3 years who did not receive the treatment, with year-on-year increase from 0 to 11% to 26% of children having lost the ability to walk and only 9% have received treatment who lost ability to walk. And again, along similar lines as the data for the respiratory function on the etapilton, now data 4 years suggest that 3 years suggest that the annual decline has been slowed down by approximately half on this patient. And our data on efficacy, and both from a protein perspective, but also clinical perspective, have been confirmed in a separate study with a similar but not identical molecule developed by the NF Pharma company. And you will recognize that from baseline, also this patient had an unequivocal increase in dystrophin expression using different technologies to measure dystrophin expression, but a clear expression also using immunocytochemistry. You can see on the top right the level of dystrophin in at least one of these patients. And also with suggestion of this is a shorter term study, was only 25 weeks, and with some differences in the slope of decline in the patient who received the treatment compared to the patient who did not receive the treatment. So I think all in all, there appears to be some efficacy for this morpholino. Of course, we all would like stronger drugs. Morpholino are very well tolerated. They really give no significant side effect apart from the fact that they need to be given repeatedly. But clearly, we would like something that is even stronger. And his development, both in the academic world, but also with the company, and in particular Sarepta now, they have an ongoing clinical trial with a morpholino that is modified with a peptide that increase the cell penetration and increase the level of dystrophin. This slide gives indication of how this particular modified morpholino works in animal models. In the human, we do not know today, but we will hear next year. And hopefully the data shows that the safety of the compound remains good, but that the level of dystrophin is higher compared to what we have demonstrated so far with the current generation of morpholino antisense. I think what many of you now would be wanting to hear is what's happening in the field of gene therapy. And if you like, the field of gene therapy is not even progressing fast, is exploding. And it took a long time to get where we are. The reason being that it has been difficult to think how to squeeze the large gene for muscular dystrophy into an AAV vector. This AAV they cannot accommodate large genes. And just to give an idea, the gene for muscular dystrophy is more than 2.5 times bigger of the maximum size you could put inside. However, experience and ideas that was generated by looking or finding that some rare families with Becker muscular dystrophy have large part, sometimes 50% of the gene missing, led to develop some shortened microdystrophin that may not be as good as the completely normal dystrophin, but at the same time gives a very significant protection of muscle from progressive damage. And now a number of companies, Sarepta in collaboration with the Nationwide Children's Hospital, Solid Bioscience, and Pfizer in collaboration with Bamboo, have developed they have gene therapy trials ongoing. And very recently, Genethon was also given the go-ahead for starting a gene therapy program in France. And I will be involved when this trial arrives in the UK. The first data to be published, I have been the one from Jerry Mendel. And these a small group of children, these were the first of the children who received, if you like, in a what we call an open label trial, receive this medicinal product. These were just 4 children. And you can see on the top of this slide the children receiving, sorry, the baseline biopsy of these children, and on the bottom of the slide, the dystrophin. And this is clearly very different from what I showed you before for antisense oligonucleotides, with a more than 80% of dystrophin positive fibers, and also with high level of dystrophin on the Western blot, that is this middle band that you can see in these gels, with levels that really go between probably 60, 70% and 90, 100% of what could be the level of a normal dystrophin. But more importantly, there is some sign of, again, these are early days. This is the first 4 patients treated for a period of a year. And on the left-hand side, you have patient 1, 2, 3, and 4. Then the parameter, NSAA, stands for North Star, and then the CK level. And then you have the baseline and the values at different time points. And I would just ask you to concentrate on baseline North Star and CK level on patient 1, for example. And what you can find that the level at baseline of North Star is 18 points, and after a year, 25 points, with an improvement of 7 points, and a decrease of CK of 46%. And I think you have this information for every single patient. So I think that there is, sign of activity. There are some difference. Some patients appear to respond better, but these are only 4 patients. And I think this is encouraging. And on the whole, the treatment in this patient was well tolerated. Pfizer is another company that has got quite a large studies underway. And this is a study where 2 different doses of AAV are given. Each of these AAV is relatively similar, but they're not identical. And they have reported at meetings, this is the data were reported at the Muscular Dystrophy Association earlier on this year. So these were a number of patients, and you have on the bottom, on the top, the pre-treatment biopsy, on the bottom, the post-treatment biopsy with clear and unequivocal dystrophin expression at quite high level. And this slide is a little difficult to understand without a pointer, but if you concentrate on the middle part, where you have this kind of spaghetti plot in light gray, then you have in dark black a line that is a kind of curve aiming towards the bottom. That curve is the average of clinical trajectories of people with Duchenne muscular dystrophy who do not receive experimental therapies. And in red, you have the trajectory of the 2 patients who appear to diverge from what would have been expected for their condition and appear to demonstrate some improvement. Also in this study, CK had been reduced. And now a number of patients, I don't even know exactly how many patients have been treated so far, but these were the first 2 patients of a much larger cohort that has been studied. As always happens, new therapies can lead to new unexpected finding, and there has been a few of the patients in this trial who had relatively mild problems, such as nausea, vomiting, that however required hospitalization. But in one particular patient, there had been some renal injury related to an immune aspect, an immune problem related to the AAV. And I think this was, did resolve without any long-term problem for this patient, but clearly created some anxiety for this patient at that particular point in time. And I think that a similar problem, but even more significant, have been observed in a separate study from Solid Bioscience. This is Ignite DMD, where also 2 different doses of AAV are being tried. And And in this study, if like the patient in the who have received the higher dose demonstrated a clear expression of dystrophin. Sorry, they are trying to call from EMA, so it's not a personal call. And and I think that there is a clear expression of dystrophin in this patient. But again, there have been adverse events in this clinical trial with that led to a prolonged interruption of the clinical trial. But this was more recently restarted because Solid was able to identify a way to mitigate some of the problem related to adverse neurological problem experiencing in the patient in the clinical trial. So hopefully we will see soon the restart of this trial, and hopefully this we will not experience, or the colleagues at Solid will not experience any further problem. I think that I suppose this is one of my last slides, is only to say that people like me, and there are now a number of people like me, are being trained on how to consider to administer these drugs, both in terms of the practicalities of administering this drug, and there is no commercial product for Duchenne, but we all started to be ready for the AAV commercial product for spinal muscular atrophy. And at the same time, and the same occurs for spinal muscular atrophy, I think that it's important that we understand some of these adverse events. This is a list of adverse events that little by little have been experienced. This is a beginning of a journey, so it's not unexpected that there are adverse event that for which there was relatively little knowledge before. And I think it's important that we have strategies to deal with this adverse event in a way that is satisfactory. And I think that there are aspects of AAV gene therapy that continue to be, if you like, need to be resolve in the longer term. That is, how long will this AAV be there for? Could this be a treatment forever? I think this is an issue that at the moment nobody really has a final answer. And of course, this leads, if you read just the lower part of the slide, leads to question regarding the previous topic was on newborn screening. So if there was newborn screening for Duchenne, should we treat a newborn? Or is there a risk that because of the muscle growth that is treated the child a little too early, and the AAV gene therapy may have not be as present as desirable after several years? Or should we wait a few years? These are important questions. It's a good question to have, but they are questions for which we will need to to identify a solution. So these are very dynamic times. I think that for us, for spinal muscular atrophy, Duchenne is also experiencing now a really acceleration of therapeutic development. At the same time, I hope you also appreciate that we still need to understand how all this treatment work. I make the point of antisense, the next generation antisense AAV, that is very exciting, but also brings new aspect related to management of adverse event. And I think this is an important journey to do properly so that we have safe and effective drugs for our children in the future. This is my last slide, and it's just to thank the people who work with me, and thank you for your attention. And this is my last slide.
Thank you very much. I have just one question. Why the virus vector could be injected only once?
It is because this is this is how we wish to get COVID vaccination, so that we don't get COVID, because our body recognize if we were going to get COVID, after a few months, our body recognizes, then destroys COVID before it becomes a problem for us. That's how we get vaccinated for mumps. But in this case, if we did get the AAV for dystrophin, we would be vaccinated against that vector, and our body would not be very happy to receive the same again. And we would be, so to some extent, this is how vaccination works.
Okay, thank you very much. I'm sorry that we haven't more time for discussion. It is very interesting, and the Duchenne disease, I think, should be observed as a future also for some new neurological maybe indications. Thank you very much, and I will.